coming to attend this seminar. Our today's seminar speaker is Dr. Ghosh, Amalendu Ghosh. Dr. Ghosh came from Washington State University. He's a Fulbright scholar there, and he is conducting research on Thrips and orthotospovirus relationship. Dr. Ghosh was born and brought up in India. He achieved his PhD degree from Bidhan Chandra Krishi Vishwavidyalaya, Bidhan Chandra Agriculture University. And he did that in 2010. He is basically an entomologist. There is both master's and PhD is in entomology. They are in entomology. Dr. Ghosh currently works as a scientist in the Advanced Center for Plant Biology in ICAR, Indian Agriculture Research Institute. His main focus is to understand the relationship between thrips and orthotospovirus that affect our horticultural crops. Mainly, his approach is to stop transmission of virus by genetically engineering method, molecular method. So this is very important, as you know that thrips transmit orthotospovirus. They have two kinds of damage: one is feeding damage, and also indirect damage, which is by transmitting orthotospovirus. Feeding damage you can stop, or you can kill some insect by using insecticide, but orthotospovirus you cannot stop that. So this is one of the most important techniques in addressing that problem. Dr. Ghosh also demonstrated the inhibition of transmission of orthotospovirus by thrips and whitefly. He has a lot of publications in that line, and he showed that it can be possible by molecular method. He is a very bright scientist in this field. He is a recipient of several distinctions. He is a recipient of a scholarship, very prestigious fellowship, which is given by the Australian government. He also recipient of young scientist scholarship. Dr. Ghosh has won a lot of competitive grants, very competitive grants. And now here is Dr. Ghosh, and he will tell us about the story of his research. So I invite Dr. Ghosh to come and give his thoughts.
Thank you, Professor Steven, for the nice good words for me and inviting me in the TREC to showcase talk about my research activities and also visit this so beautiful place and meeting new people. I work basically on thrips and orthotospovirus as a senior scientist at Indian Agricultural Research Institute, New Delhi. And right now I'm visiting Washington State University under Fulbright program. Before I start, just to say from where I am, like in this direction.
Ready? Try the other button.
Yeah. So yeah, I saw lots of scholars here from India, so I'm not talking more. So I'm from the capital region of India, Delhi, which is famous for its iconic historical monuments and also very tastier and delicious food. and at the same time, it's too populated in Delhi. And our institute, IRI, is in the heart of the Delhi. So its abbreviation is IARI, Indian Agricultural Research Institute, which is established in 1905 and then came to Delhi in 1936, and it's the seat of Green Revolution in India during 1970s. And right now it ranked first as the agricultural and allied university in India. And yeah, few of our alumni, Aru is from there, Sumit is also, has some connection, So, we have our world-renowned alumni who got World Food Prize, starting from the late Dr. Saminathan, and Sanjay Rajaram, Basal, and recently Dr. Rajan were alumni of our institute. Yeah, Delhi is highly populated, but if you enter IRA, you will forget that you are in a so populated city. So IRA is luster of greens, and these are some of the glimpses. This is our library view. These are our experimental fields, and we have 1,500 hectares area within the city of Delhi, in the heart of Delhi, including our experimental field and the office premises. And right now we have 6 schools where you come as a college within the institute. So like crop improvement, plant protection, basic science, natural resource, then social science and horticultural science. So we come under the School of Plant Protection that has like 4 departments: nematology, agricultural chemicals, pathology, and entomology. And total, we have 20 departments, 8 research units, 1 project direct, 8 regional stations across India, 2 regional centers, 1 farm science center that we say APK, and 2 recently we started 2 academic campus in the eastern region of India, eastern and the northeast. So now coming to my division, that is the Department of Plant Pathology. And in the plant pathology, we are working 27 scientists. And we have sections like fungal pathology, mycology or the fungal taxonomy, bacteriology, plant bacteriology. And I am working in the Advanced Center of Plant Biology, this building, where we have 9 scientists and their labs. Oh, this is my laboratory in the virology section. So we say insect vector laboratory. So these are the current fellows working in that laboratory. Now coming to my research topic. So my lab basically works on Thrips transmission of prospoviruses and whitefly transmission of the adenoviruses. Both these pests are the insect vector of other than this plant virus group, and that causes more than 50 to 70% losses in mostly in the vegetable crop. It is havoc in the ornamental crops, although not really very good way documented. And the current practices are basically on the pesticide molecules. So I was just seeing around $420 million pesticides are being used to manage the menace of this prospovirus and whitefly adenovirus. But surprisingly, I was discussing with Ghosh, still the same situation is going on here, because these 2 pests are so quickly develop resistance against the recent chemical molecules that very difficult to control with the pest. So we started working to find out some sustainable solution that can interfere in the Thrips prospovirus and whitefly adenovirus interaction. And our target to achieve 2-fold benefits. One is reducing the crop damage by Thrips and whitefly, and also to restrict the spread of the viruses, so we get the escape from virus infection also. Okay, so this is the Thrips as a pest, and this is our Thrips we are working. So that this is a Thrips farming in our place. So in India, we say this is the predominant Thrips species for spreading prospoviruses. And I was surprised to know that this is also the one of the predominant pests right now in Florida. So basically, if you see the thrips, around 6,460 species are there, and most of the coming from this group, which are the crop pests. But it's true that majority of them are the scavenger and the pollinators. So basically, they are the beneficial one. They are not the pest, but we know them as mostly we know them as a pest. And only 1% of them out of these 6,000 species, only 1% of them are recorded as agricultural pest. And they have the phytophagous behavior. These species, they have the asymmetrical mouth parts, they wraps the exoskeleton of the cell and imbibe the liquid plant sap out of the cell. So the plant tissue become empty. And in this empty space, air entered. And due to that empty space, you will find some silvery appearance on the damaged plant parts. So gradually, this silvery appearance turns to yellow, like this one, and then turns to a little bit brown, and they develop scared, malformed food. Okay, and those are not marketable. That reduce also the yield and the quantity. And some trees, they usually name gods in the trees. So those are different groups we say wall making trees. So just before the lecture, I was updating the information. So beside their damage, hitting damage as a pest, they transfer a lot of plant viruses. So far, up to date, we recorded around 29 tospoviruses. We say tospoviruses, but recently the genera, they redefined the genera and the new name is orthotospovirus. But now tospovirus is widely accepted for a long time. So while I'm speaking, I will tell tospovirus only. So 29 tospoviruses, 3 ELAR viruses, and one species each for carmovirus, macromovirus, and the solenovirus. And for this much of virus, there is only 19 Thrips species so far is recorded. But this 19 is making so much damage across the world. So among these 19, I put red letter for 2 species. One is Frankinella occidentalis, which is the #1 Thrips pest in US, in the Western world, in US and Europe also. And Thrips palmi, we consider this is the brother of Frankinella occidentalis in the Asian countries. So this is the predominant species. You also see the number of viruses transmit, the list is bigger for 2 of them. And TSWV, we said the type species of tospovirus, that is the tomato spotted wilt virus, that causes devastated loss in many provinces of US since decades. So TSWV is one of the important tospovirus that causing $1 billion loss every year. So if you see the epidemic of tospoviruses, so first it was identified in 1905 from Africa, but that time it was not designated as like TSWV. They said tobacco wilt disease. Later on in the 1915, they described the causal organism tomato spotted wilt virus in the tomato. But that time, predominantly it was transmitted by other Thrips species like Thrips tabaci, and later on people added Frankinella sulci. Frankinella occidentalis was not there. And that time there was no epidemics. It was just first report or something. So epidemic of the tospoviruses started post 1980s, this period. And it was noticed that every outbreak was followed the introduction of Frankinella occidentalis at that time. So it's in the America, then in the Europe, and also in the Australia and Africa. So yeah, so we say Frankinella occidentalis is the super spreader of the tospoviruses. Okay, because every epidemic was caused by coexistence of Frankinella occidentalis and the TSWV. And up to 1990s, there was only one tospovirus species, that is the TSWV. In 1990s, post-1990s, many other sister species coming reported, like INSB and then TLCZ and JHCV. CSCV also came, and also with that, many new vector was started reporting. If you see the Indian scenario, that is our condition, or you better we see the Southeastern Asia, like in Indian subcontinent, and also including the Chinese province also. So in 1960, we thought reported some phosphovirus-like disease, but we are not sure what was that. And it took long time because it was not havoc, not causing damage, so people did not pay attention. So it was the past in 1990s when it was identified it is not the TSWB like in the Western countries. So it's a different virus. And people name it groundnut bar necrosis virus or peanut bar necrosis virus. So we attributed this either GBND or PBND. And also later on within a year, another virus reported that is the watermelon bar necrosis virus, WBND. So it's infecting all the melons and other cucumbers. Later on, another capsicum produces virus is reported, and all are transmitted by Thrips palmi and other species also like IYSV is there in onion and garlic, ID yellow spot virus, so that is fully transmitted by tabas. And yeah, so Thrips palmi, if you see the distribution map, it was restricted to the East Asia and Southeast Asia up to 1990. And in 1989, it first came in the US also. It started spreading the US, Caribbean, and the South America. And in US, it is predominant on the southern provinces. I'm working on in the Washington state, and I don't see any farming there. So, yeah, if people can say that, yeah, it came from India or yeah, you are the agent like carrying that to America, but at the same time, we feel that Frankinella occidentalis, which was not present at all in the Asian countries, in the South Asian countries, we got first incidence of Frankinella occidentalis just 3 years back. And with that, we got also the tomato spotted wilt virus that was also not present in India earlier. So in 2018 and 2019, we first report coexistence of Frankinella occidentalis and the TSWB both in Indian subcontinent. So probably this is the changing climatic condition. So the pest which was confined to the tropical countries in Asia and the pest which was confined to the Western world, now with the changing climatic condition, they are expanding their adaptation and establishing in new places. So we got Frankinella occidentalis from Western world that established in the Asian countries also. And now from the, which was this predominant, like Swami, it was this predominant in the Southeast Asia. Now it is expanding its adaptation to the America and Europe. Still, they claim they are free from Thrips farming and they listed it as a quarantine pest. But in few nations, they already reported the existence and they started eradication process for Thrips farming. So what is the economic significance? I thought little bit for TSWB, we recorded around 1 million loss. And in US data, I was seeing in the last 10 years, there is 1.4 billion losses due to the phosphoviruses and thrips. In India, only for GBNB, including the other countries in the Indian subcontinent, around 89 million losses are there. Only due to one virus, and WB also like 50 million was recorded. And WB is so devastating in the cucumber crops that people left the cultivation of watermelon and other melon due to the infection of the watermelon bud necrosis virus. So our lab started working on this PASO system because whatever information is available, as this virus complex is well established in different nations, people work on this one and little bit of Frankinella sulci. There was very limited information on that virus vector relationship on Thrips farming and median with WBNB, which is predominant in our condition. So our lab started working on that with the aim to interrupt the interaction by different means and to save the crop loss. So this is the biology of our species. So they have egg stage. So egg stage are invisible, like 50 micron in size. You can't see them in the naked eyes, and they insert the eggs within the plant tissue. So those are totally invisible. And then from the plant tissue, the larval one, we say L1, that comes out. And they have another larval stage, we say L2. And then they have two pupal stage, P1 and P2. And they are adult, like male adult and female adult. This one. So the interesting fact about the virus vector relationship is that only this L1 stage, the first instar larva, they can only acquire the virus. And the pupa, they are completely non-feeding stage, or little bit they move sessile stage mostly. So they don't have any role either in the acquisition of the virus or inoculation of the virus in the new plant. So who are inoculating? So only the adult stage, they inoculate the virus to the healthy plant. And they become viviparous only when the larva acquire the virus. If adult acquire the virus, they cannot become viviparous. This is very interesting. So we try to know why it is so, why adult cannot become viviparous if they are taking the virus in that stage only. So they can become viviparous only the acquisition is in this stage. So that we answered. And also, if you see, when the Tossovirus is acquired by the Thrips, that has some pathological effect on Thrips biology also. So that reduce their survivability, reduce the reproduction capacity, but they modify the life cycle in so nice way that favors the spread of the virus in higher magnitude. So this is the stage for the virus acquisition. But if they acquire the virus, this stage is little bit extended. So that favors them to acquire the virus more efficiently and become viviparous. So this is the modification in the relation that virus is conferring. Now coming to that transmission again, I was taking that why they cannot become viviparous. So this is the schematic diagram of Thrips elementary canal. So their elementary canal is big tubular. So we say this is the foregut, then they have so much long midgut. And the midgut has three positions like midgut 1, midgut 2, and midgut 3. They have multigenital and then they have hindgut. And this is their salivary gland. So this is the main salivary gland. We call it primary salivary gland or PSG. And they have an elongated projection that is called tubular salivary gland or TSG. So if you see the diagram, you will understand the salivary gland is directly in physical contact with the midgut through this tubular salivary gland. And also they are connecting to a pair of ligament, this LG. So that also connects the anterior midgut to the salivary gland. So what we know from the TSWB and the Frankinella oxygendalis interaction, the group like Goodman group is working for a long time, Whitfield is working for a long time in the US. So they said that the virus directly travels from the midgut to the primary salivary gland through this connecting PSG. Because for successful transmission, the PSG should be infected because the virus comes out of the larval body through saliva when they feed on the healthy plant. So necessarily the salivary gland should be infected for successful transmission. But when they are taking the virus come to the alimentary canal with the food material and the midgut is infected. And from midgut, they said it travels to the salivary gland and reaches the primary salivary gland for successful infection. So we tried to understand whether the phenomena is sponsored across the Thrips family or this thing, other Thrips family and Tossovirus. So we worked on that. And yeah, we dissected the minute Thrips, every stages of life stages of the minute Thrips, and you will see this is the larval picture and this is the adult picture. So what I was talking about in the schematic diagram, this is the connecting ligament, one pair, and this is the tubular salivary gland. So it is coming from the primary salivary gland and touching the midgut. And both this connection, the ligament and the tubular salivary gland, this is adult picture. So this is intact across the life stages. So what they said, it breaks and they cannot transmit that in the adult stage. So earlier it was told like that they cannot become viviparous because, the TSG, the connection of TSG breaks in the adult stage. But we demonstrated that the connection is intact throughout the life stages. So the understanding that adult cannot become beautifiers because the TSG connection breaks, that is not correct for at least for our Thrips and Tossovirus complex. Then we try to identify then what is the root of the virus infection. So that took long time to establish, and yeah, this is like we tag the virus particle with a green fluorescence and the actin filament present in the elementary tract with the red fluorescence spotting. And you will find where they are merges, this picture is merge of A and B panel. So where they are merges, you will get a little bit oranges to yellow fluorescence. So the first infection, it's the L1 larval stage 1 in elementary canal, and the first infection they received in the anterior part of the midgut. And this is the enlarged person. You will find the circular epithelial cells in the midgut of the Thrips palmi. And within the circular epithelial cell, green fluorescence is coming out. So those are the virus infections that is tagged with the virus encoding. So that proves that first infection comes in the epithelial cell of the anterior midgut. And immediately after that, we found the infection in the connecting ligament. So from the anterior portion of the midgut, the infection is transmitted to the connecting ligament. And from ligament, you will find immediately after that, the primary salivary gland is infected. So we concluded that, so it's not the TSG, it's the connecting ligament that helps, that is the channel of the virus dissemination from midgut to the salivary gland. Because at that stage, the tubular salivary gland was not infected, so it cannot be the primary channel. Okay, so with that, and another thing is that we tested the hemolymph, that is the blood of the insect. There is no virus present. That is also a unique feature because it's most of the insect and virus interaction. Hemolymph is the route to transmit the virus from elementary canal to the primary salivary gland. But in case the virus is not routed through the insect blood, it is routing through the connecting ligament. And yeah, TSG, we got infection also in the TSG, but it is in the later stage of the development. So maybe they have a secondary role in the virus infection route, or maybe the infection is TSG is just the reverse infection that is coming from the PSG to TSG in the reverse direction, because TSG is also connected with the PSG. And in the adult stage, we found that there is no infection in the epithelial cell, unlike the larval stage. Larval stage, the infection was only in the epithelial cell. In adult stage, you will find a lot of longitudinal and transverse line. So those are the muscles, longitudinal muscles and the visceral circular muscle of the elementary tract. So that once the infection is there, so that infection transmitted to the visceral circular muscle and the longitudinal muscles during the adult stage. So the infection is retained there. In fact, when we are feeding to the adult, we don't see any infection in the epithelial cell. So basically, epithelial cell, the susceptibility of the epithelial cell, that determines the fluviferous nature of the Thrips. So epithelial cell in the adult are not susceptible to tossovirus infection. So that is the reason why the adult is acquiring the virus, they cannot become fluviferous. So it can become fluviferous only epithelial cells are infected in the larval stage, and in the adult stage, the same infection is retained in the muscle cell of the adults. And yeah, this was probably the first evidence of this phenomenon in Thrips, and specifically the Thrips family we worked on. Yeah, and this is another thing we looked at, what is the interaction going in the cellular tract. So this is the first time we developed a cell line out of scratch for Thrips farming. Sumit was associated with this work. We developed a primary cell culture of the Thrips. From the different, these are the studies done by Sumit, and he studied the different embryonic stages of the Thrips, and we identified this stage around 60 to 70 hours that are good to develop the, this type of primary cell culture. from the embryonic tissue. And then Sumit, this part was totally done by Sumit, and we infected the primary cell culture with the purified virus study, and we said the virus N protein is localized in the nuclei of the cells. Those are multinucleate cells, and this is probably the first evidence where earlier understanding was that the Tospovirus infection is restricted in the cytoplasm. But this is the first evidence where we could see that infection is moving to the nuclei of the cell. So this cell culture is very much important to study the in vitro interaction of the viral protein with the Thrips protein. And yeah, we are yet to work on that to make it a permanent cell line and make it bring it up to the market level. And yeah, then we are looking for the targets, what are the receptor of the virus in the Thrips cell. And first we started with some computational analysis and we find syntaxin 6 and some ubiquitin expression protein that actually interacts with the Tospovirus gene and helps in the internalization of the virus particle. And that followed by transcriptome, and those rates were also very high in the Thrips transcriptome post virus infection. And then we went for those genes to silence them and see their expression and also their function in the virus transmission. So this is the one approach in the silencing study. So that we undertook. So this is not the conventional way of the silencing. So here we used the modified antisense oligos. So that is activated as ASO. So in ASO that acts in a defined manner that they goes with the RNase H-mediated degradation of the target mRNA, and also they hinder the translation, and also they do the mistranslation. So this was very much active than the conventional dsRNA we compared for the same target. And but the disadvantage people don't follow this technique because these are small RNA oligos and these are highly susceptible to the nuclease activity. So that is very ancient classical technique, but people don't adapt it for the balancing experiment because of their instability. So what we did, we made some modification in the backbone of the oligos. Basically, we did the H modification in the 3' position, and that helps nuclear stability and also that increase the penetration of the oligos in the tissues. So then problem is that this is so tiny insect and very difficult to deliver your target molecule in the fish. And these are sucking insects. It's not like we cannot spray on the surface, they will not pick up because they suck the plant suck on the tissue by inserting their snout stylet. So this improvisation was made. So we just modified one kind of tube for the feeding apparatus and put some liquid dye for them and covered it with the paraffin. So what they do, they sit on that and they fires the paraffin cake and started sucking the liquid dye. But still we are confused that whether they are picking up or not, how do we understand? So we add one non-toxic dye. So if they feed a blue stinge coming in their alimentary canal and their abdomen is swelling, so we can see them from outside and confirm they are eating, they are drinking the liquid dye. So basically we targeted one of the ubiquitin expression protein and one of the immune virus immune protein. So expressing gene. So this we tried and yeah, this has good effect on their virus acquisition. So virus acquisition was lowered by around 200 to 300 times post the application of the antisense oligo. And that we did not expect that because they create a lot of changes in their physiological process. So that induces a high mortality in the treated Thrips. And that creates the morphological deformities also. When we the UHRA binding protein 1, we silenced, the body became flaccid and abdomen got flattened. in case of EFAS, the appendage is twisted little bit, and insect become very brittle, and they die. But nothing is like in the control panel. So this we targeted to this one like ubiquitin expressing protein that we could further see. Yeah, morning I just added a few another studies. So Rajesh is there. Rajesh worked with me. He completed masters at IRI. He is from the University of Georgia, now doing PhD there. So he did some of the silencing in Thrips again with some BTK 29 and then collagen, some of the collagen formula. So that also has a little bit suppression of the virus transmission. But if we silence the collagen in Thrips, the virus titer in the Thrips increased. So basically, collagen is associated with antiviral immune response. So if we eliminate the collagen expression, the virus concentration started increasing in Thrips. But now we are looking for to take this tyrosine kinase in Thrips for further assessment. So with this background, now we started working on the editing the ubiquitin expressing gene, like with associated with UHR binding protein 1, we targeted the ubiquitin lipase, that is the EBR7 in Thrips, and this is the classical pathway of the Cas9 based gene editing. So we are synthesizing the single guide RNA and then administer them in the Thrips. So basically, this study we started back at IRI and then carried forward at WSU Pullman. So in WSU Pullman, so far what we got that, in case of, so other than this, we took two controls. So one is yellow protein and one is the white protein. So the purpose of taking those control is yellow protein is responsible for body melanization that gives a yellow color to the Thrips body, and white protein that gives the eye color of Thrips. So if we silence them, the eye color and the body color will change if we edit them. So by seeing them phenotypically, we can say that our construct are working. So that was the purpose of taking yellow protein and white protein. And ubiquitin ligase is associated with the orthovirus transmission. So that is the purpose of targeting them. So so in the in vitro study so far, the RNP complex that comprising the Cas9 protein and the single guide RNA that efficiently cleave a 1.2 kb linear product of the white protein into two parts. Like this one is 980 dt and this is 3020 dt. And same happened for this is not the multiplex, this is the single plex, sorry. And here for the yellow protein is around 1 kb linear product is split into two parts when we are administering the RNP complex of mixing the Cas9 and the single guide RNA into 450 and 550 dt. And in case of the ubiquitin expressing gene, so here we have two single guided RNA. This is one set and this is the other set, and this is the control panel. So here you can see it is the 2 kb or 1.2 kb. 1.1 kb. That is the linear product, but when we are administering them with the protein complex of the Cas9 and the single guide RNA, that leave the linear product into 750 and 350 dt, and there it is around it's around 650 and 450 dt. And if we mix them, so in the multiplex also they are working nice. So they will chop the target linear product into four parts. So this is a 1.1 kb product, and that is cleaved into 750, 650, 450, and 350 kb product. So now what we are working on to develop a mutant population of the Thrips that can incapable of the virus transmission, and also they have like mutated white protein and the yellow protein gene that will change their eye color and the body color. But again, like I said, those Thrips, we have to do the injection or delivery of the RNA complex in the single cell position of the Thrip. So that is tricky because we need to get the Thrips embryos, and the Thrips eggs are inserted within the leaf tissue. So those are invisible. So if we want to inject, we have to get them. So this is the improvisation we made. So this is like a petri plate, and within the petri plate, we provide a little bit of pollen. So pollen are very much help to give higher production of embryo and Thrips. And then we put a bathroom tape here, and again on the bathroom tape, we have sterile water, and we sandwich the sterile water with another layer of bathroom. So what they do, they have a very soft ovipositor. So they insert their ovipositor with the bathroom tape and lay the eggs on in the water sandwich between them. Then we have to collect it, and we under microscope we see the cellular stages of them. So getting single cell is very difficult, but at least we are trying. We should be limited up to 4 cell stage of the embryo. And in the 1 to 4 cell stage, we are micro-injecting the RNP complex in the Thrips embryo. So this is looking pretty big, but this size is like 20 microns, something like that. So I can't see in eyes. So under microscope, and this seeding size is 1 micron. So yeah, putting 10 micron, putting the substance in a 10 micron size with 1 micron, less than 1 micron, its length is around 50 micron and width is around 5 to 10 microns. So we are pushing all the construct within that and developing that muted population. So we are still under this stage to develop the muted population and go for the further analysis. And yeah, I hope before I leave US, some of the study I can conclude here. And with this, I just finish my talk. And yeah, I acknowledge the support I received from the Fulbright Scholar Program. My lab, this is my institute and my organization, Indian Council of Agricultural Research. My lab is sponsored by 2 ongoing projects of the DVT and Science and Engineering Research Board. And then we have another sponsorship from DVT Northeast Development Program. One program is going with the Australian government and India collaboration. So that is also funded by the project from Australian government. And Washington State for hosting me. This is my group in the Washington State. This is my supervisor, Professor Anupatu, very nice gentleman and very generous and supportive. This is my lab photos in 2021, 22, and this is the kind group I am missing because I am not there. So yeah, you will see some known faces. Sumit, yeah, long time was with us. So Sumit is there in 2001 and 2002. And yeah, Rajesh, your picture is there?
Yeah.
Video?
Yeah.
Somebody is there, so he is not also there. And then, yeah, 2 similar pairs I just detected. Sirsa, he was also with us. Now she is in the same lab with me in the Washington State. And Manish was about to meet me there. Manish is also from, Manish is there? First picture. Yeah, he is Manish. So Manish is also in the Tipton campus, Georgia. So he's working with Dr. Bag. And yeah, we missed him this time. And yeah, special thanks to Dr. Steele, who actually hosted me for this outreach lecture fund sponsored by the Fulbright. And yeah, I was a long time I was following this publication and work because I find the Thrips family who is working in the US, so only one publication found, that is the one group publication found, that is from Professor Steele. So it's so nice of you to invite me and hosting me for this lecture.